Hello, everyone, and welcome to today's fascinating journey into the world of mycorrhiza. In this video, we will delve deep into the captivating symbiotic relationships between plants and fungi that have a profound impact on our ecosystems and agriculture. Mycorrhiza is a topic that bridges the gap between biology and ecology, offering insights into how plants thrive, adapt, and even communicate beneath the soil's surface. Whether you're a student exploring the wonders of the natural world or a gardening enthusiast looking to boost your plant's health, understanding mycorrhiza is a key piece of the puzzle. We will explore the different types of mycorrhizal associations, the incredible mechanisms at play, and the vital role they play in nutrient exchange. What is mycorrhiza? Mycorrhiza refers to the symbiotic association between specific fungi and plant roots, where the fungus aids in nutrient and water uptake for the plant, and in return, the plant provides the fungus with essential nutrients produced through photosynthesis. Mycorrhiza, derived from the Greek words mucophyta meaning fungus and zeta alpha, rhiza, signifying root, refers to the symbiotic association between specific fungi and plant roots. This relationship is predominantly mutualistic, where both organisms derive benefits. The fungus aids the plant by enhancing water and mineral ion uptake, thanks to its extensive hyphal network that provides a broader surface area for absorption from the soil. Conversely, the plant supplies the fungus with essential nutrients, particularly sugars and nitrogenous compounds, produced through photosynthesis. There are two primary modes of fungal colonization in a mycorrhizal relationship. In the case of arbuscular mycorrhizal fungi, the colonization is intracellular, where the fungus penetrates the root cells. On the other hand, ectomyocorisal fungi establish an extracellular association, enveloping the root tissues without intruding the cells. The nature of this association can vary based on the species involved and environmental conditions. While many mycorrhizal relationships are mutualistic, certain scenarios or specific species might lead to a parasitic interaction, where one organism benefits at the expense of the other. It's noteworthy that not all plants form mycorrhizal associations. In regions where the soil is rich in water and nutrients, plants might not necessitate the services of mycorrhizal fungi. Similarly, under such conditions, mycorrhizal fungi might not find it conducive to germinate and proliferate. However, for certain plants, the presence of mycorrhizae is crucial. A quintessential example is the pinus species, where mycorrhizae play an indispensable role in seed germination and establishment. In the broader context of ecology, mycorrhizae significantly influence nutrient cycles, impacting plant evolution, physiology, and overall soil dynamics. By facilitating nutrient exchange and enhancing soil biology, these symbiotic relationships underscore the intricate interdependencies in terrestrial ecosystems. Types of mycorrhiza. Mycorrhizas are often classified as either ectomycorrhizas or endomycorrhizas. The hyphae of ectomycorrhizal fungi do not penetrate individual root cells, but the hyphae of endomycorrhizal fungi invade the cell membrane and pierce the cell wall. Endomycorrhiza consists of arbuscular, ericoid, and orchid mycorrhiza, whereas ectoendomycorrhiza comprises arbutoid mycorrhiza. Monotropoid mycorrhizas constitute a unique category. Ectomycorrhiza. Ectomycorrhiza, ECM, represents a specific type of symbiotic association primarily found between certain fungi and the roots of woody plants. Notably, tree genera such as birch, beech, willow, oak, pine, spruce, ephyra, and eucalyptus are involved in these relationships. This association is characterized by the formation of a heartig net, an intricate intercellular network of hyphae that interfaces with the epidermal and cortical cells of the plant root. Externally, a dense sheath of hyphae, termed the mantle, envelopes the root surface, distinguishing ectomycorrhizae from other mycorrhizal types. Intriguingly, ectomycorrhizae do not penetrate the root cells but remain external, establishing a significant portion of their fungal biomass outside the root. This external structure comprises two main components, the aforementioned mantle and the extraradical hyphae that extend into the surrounding soil. The mantle can vary in its complexity, ranging from a simple network of hyphae to a multilayered structure resembling plant parenchyma tissue, known as pseudoparenchymatous. The heartig net, situated internally to the root but external to the cortical cells, serves as the primary site for nutrient and carbon exchange between the symbiotic partners. The colonization by ectomycorrhizal fungi often leads to suppressed root hair development and induces changes in root branching patterns. These alterations can range from simple bifurcations to extensive branching, resulting in multiple root tips enveloped by a continuous hyphal sheath. Such extensive branching is sometimes referred to as coralloid or tuberculate. The fungal hyphae also impart a distinct coloration to the mycorrhizal root surface, aiding in its identification. Ectomycorrhizal fungi, which include members of the Basidiomycota, Ascomycota, and Zygomycota, are often associated with forest and woodland mushrooms such as Russula, Hebeloma, and Amnita, as well as truffles like tuber. While many of these fungi are generalists, associating with multiple tree species, some exhibit specificity, forming symbiotic relationships with only particular tree genera. A unique variant of ectomycorrhiza, termed ectendomycorrhiza, exhibits characteristics of both ectomycorrhizae and arbuscular mycorrhizae. This association is particularly observed in pines and larches and is attributed to specific fungi like those from the Pezizales order. In summary, ectomycorrhizae play a pivotal role in terrestrial ecosystems, facilitating nutrient exchange and enhancing the survival and growth of their host plants. Their intricate structures and diverse fungal associations underscore the complexity and significance of these symbiotic relationships in nature. Structure of ectomycorrhiza. The structure of ectomycorrhiza, a vital symbiotic association between fungi and plant roots, consists of several key components. Heartig net. This intricate network of fungal hyphae serves as the primary interface for nutrient and carbon exchange between the fungus and plant. It is initiated by hyphae from the mantle and extends into the root, forming a crucial junction for nutrient exchange. The depth of penetration varies among plant species, and adaptations in root cells enhance surface contact. Mantle. Enveloping the root, the mantle consists of fungal hyphae and can vary in structure from loose meshes to stratified tissue systems. It can inhibit root hair development, induce hormonal responses, and facilitate nutrient exchange. Diverse mantle forms aid in identifying specific ectomycorrhizal associations. Extraradical hyphae. These hyphae extend from the mantle into the soil, compensating for the reduction in root hair development caused by the fungus. They can form rhizomorphs, complex structures with various growth patterns, and serve as conduits for nutrient transport, potentially spanning long distances. Fruiting bodies. Ectomycorrhizal fungi produce conspicuous fruiting bodies, such as mushrooms and truffles, essential for their sexual reproduction and spore dispersal. Fruiting bodies contain unique biochemical compositions, facilitate mutualistic interactions among different fungi, and are dispersed by various vectors, including animals and wind currents. These structural components of ectomycorrhiza play vital roles in nutrient exchange, plant-fungus interactions, and ecosystem dynamics, highlighting the complexity and ecological significance of this symbiotic association. Mechanism of ectomycorrhiza. Ectomycorrhiza is a specialized symbiotic association between certain fungi and plant roots, featuring intricate mechanisms that support nutrient exchange, protection, and mutual growth. The process can be summarized as follows. 1. Presymbiosis. Fungal hyphae seek plant roots and penetrate the soil. Root cap cells are infected by the fungus to establish symbiotic structures. Gene expression changes in both plant and fungus, and chemical signaling from the plant stimulates fungal growth. 2. Symbiosis. Mantle formation by fungal hyphae progressing from root cap to epidermal cells. Specific polypeptides, ectomycorizines, are produced during this phase. The mantle regulates root growth, root hair production, and branching. Heartig net formation facilitates nutrient exchange through various interfaces. 3. Non-nutritional benefits. Extraradical hyphae transport water and provide physical protection. Fungal secondary metabolites act as biochemical defenses. ECM fungi enhance plant stress tolerance in challenging environments. 4. Ectomyocorism. Some ECM fungi can also penetrate cortical cells, forming dual interactions known as ectomycorrhizas. In summary, ectomycorrhiza involves complex processes that benefit both fungi and host plants, highlighting the intricate interplay in terrestrial ecosystems. Importance of ectomycorrhiza. Ectomycorrhiza plays a significant role in various ecological, environmental, and agricultural contexts, with implications spanning multiple domains. Agricultural impact. Modern agricultural practices like tilling, excessive fertilization, and fungicide use can harm mycorrhizal health and local ecosystems, leading to changes in ectomycorrhizal communities and reduced sporocarp production due to excessive fertilization. Forestry and tree plantations. Ectomycorrhizal associations are crucial for tree growth, and in commercial forestry, introducing appropriate ECM fungi is essential for transplanting trees to new regions. This is especially important for obligate ectomycorrhizal trees like eucalyptus and pinus. Ecosystem restoration. Leveraging ECM fungi is gaining interest in ecosystem restoration efforts. Reintroducing these fungi after their loss from a habitat is vital for re-establishing vegetation and rejuvenating ecosystems. Heavy metal detoxification. Ectomycorrhizal fungi are resilient to heavy metals and can colonize contaminated soils, binding and detoxifying these metals. Fungal fruiting bodies can also concentrate heavy metals. Pollution mitigation and fluoremediation. ECM fungi demonstrate positive effects in scenarios such as salinity, radionuclide accumulation, and degradation of organic pollutants, making them valuable in pollution mitigation and fluoremediation efforts. Climate change adaptation. Ectomycorrhizal communities respond differently to climate change challenges, with elevated CO2 levels promoting fungal growth and temperature fluctuations having varied effects. Many ECM species enhance plant resilience during droughts, improving root water uptake capabilities. In summary, ECM fungi have wide-ranging ecological, agricultural, and environmental implications, highlighting their importance in addressing various challenges and contributing to the resilience and adaptability of ecosystems. Examples of ectomycorrhiza. Ectomycorrhizal fungi predominantly belong to the Basidiomycota phylum and are integral to forest ecosystems. These fungi form symbiotic relationships with various tree species, facilitating nutrient exchange and enhancing tree health. Herein, we delve into some representative examples of ectomycorrhizal fungi. Amnita spp. The Amnita genus is a quintessential example of ectomycorrhizal fungi. Notably, certain species within this genus, such as Amnita muscaria, exhibit a broad host range, forming symbiotic associations with over 20 different tree species. This non-specificity underscores the adaptability and versatility of Amnita muscaria in establishing mycorrhizal relationships with diverse tree hosts. Boletus spp. Boletus represents another pivotal genus within the ectomycorrhizal fungi realm. While some species within this genus are non-specific in their host preferences, others, like Boletus elegans, display a high degree of specificity. Boletus elegans, for instance, predominantly associates with large trees, highlighting the intricate and specialized nature of certain ectomycorizal partnerships. Tricoloma spp. Tricoloma is yet another genus that contributes to the rich diversity of ectomycorrhizal fungi in woodland ecosystems. Species within this genus are known to form symbiotic relationships with various tree species, playing a pivotal role in nutrient cycling and soil health. In conclusion, ectomycorrhizal fungi, represented by genera such as Amnita, Boletus, and Tricoloma, are paramount in forest ecosystems. Their ability to form specific or non-specific symbiotic relationships with trees accentuates their ecological significance and underscores the intricate web of interactions that underpin forest health and sustainability. Endomycorrhiza. Endomycorrhiza represents a category of mycorrhizal associations characterized by the fungal component's predominant internal localization within the root structure. This involves the fungal hyphae penetrating the host plant's cortical cell walls, establishing a close and intricate relationship. Two primary forms of endomycorrhiza are the arbuscular and ericoid mycorrhizae. However, other types, such as the dark septate endophytes, can also be classified under this category. A unifying feature across these forms is the hyphal penetration into the host cells, signifying a deep-rooted symbiotic relationship. Remarkably, endomycorrhizae are associated with the vast majority of contemporary plant species, encompassing over 80% of them. This includes a wide range of plants from agricultural crops to greenhouse flora, such as vegetables, grasses, flowers, and fruit-bearing trees. The relationship is marked by the fungi's formation of arbuscules and vesicles and its penetration into the cortical cells of the host. This internal exchange mechanism is complemented by the extension of the fungal hyphae outside the root, making it a more intimate association compared to ectomycorrhizae. Further categorization of endomycorrhizae reveals specific types, including arbuscular mycorrhizae, a widespread form known for its arbuscules, which are tree-like structures formed within the plant cells. Ericaceous mycorrhizae, associated primarily with plants in the Ericaceae family. Arbutoid mycorrhizae, found in plants of the Arbutus genus and related species. Orchidaceous mycorrhizae, specific to orchids, aiding in their nutrient uptake. In essence, endomycorrhizae play a pivotal role in plant ecology and agriculture. Their presence in a vast array of plant families, from greenhouse plants to agricultural crops, underscores their ecological significance and the intricate balance of mutualism they maintain with their host plants. Arbuscular mycorrhiza. Arbuscular mycorrhiza, AM, is a specialized symbiotic relationship between glomeromycota fungi and various vascular plants, including tropical trees. The association begins with fungal hyphae penetrating plant epidermal cells through enzymatic processes and hydrostatic pressure, forming apressoria on the root's exterior. Subsequently, hyphae navigate through cortical cell walls, creating tree-like structures called arbuscules, optimizing nutrient and carbon exchange. Two primary arbuscule types, Arum and Paris, exhibit distinct characteristics. Fungal colonization suppresses root hair development, while extraradical hyphae extend into the soil to increase the absorptive surface area. Some fungi may develop vesicles within root tissues, serving as storage units. Asexual spore production occurs both externally and within roots, aiding in species identification and dispersal through air, water, and soil organisms. Overall, AM is a vital symbiotic relationship that enhances ecological balance and nutrient exchange between specific fungi and a wide range of vascular plants. Mechanism of arbuscular mycorrhiza. Presymbiosis phase. Spore germination. AM fungal spores can germinate independently but are expedited by host root exudates. Hyphal growth. Hyphal development is influenced by root exudates and soil phosphorus levels. Host recognition. Root exudates guide aim fungal hyphae towards a suitable plant host. Aprosorium formation. Hyphae form aprosoria on the root's surface, initiating the symbiotic relationship. Symbiosis phase. Arbuscule formation. Highly branched arbuscules develop within plant roots for nutrient exchange. Host regulation. The host plant actively regulates hyphal growth and arbuscule development. Other hyphal forms, short-lived runner hyphae enhance nutrient absorption, accessing soil pores. Unique morphologies. 4 distinct forms of AMF hyphae exist for diverse functions in symbiosis. Nutrient uptake and exchange phase. Carbon exchange. AM fungi depend on the host plant for carbon, which is transferred through arbuscules or hyphae. Metabolic pathways. Hexoses are metabolized into carbon storage forms, trehalose, and glycogen. Lipid production. Lipids are produced and may be stored or converted into hexoses. Phosphorus absorption. AMF hyphae absorb phosphorus more efficiently than roots. Nitrogen flow. AM symbiosis affects soil pH, phosphorus solubility, and ammonium ion dynamics. Carbon investment. Plants invest carbon to enhance phosphorus absorption. Phosphorus absorption variability. Different AMF species vary in their phosphorus contribution to plants. Importance of arbuscular mycorrhiza. Arbuscular mycorrhizal fungi, AMF, play a crucial role in various agricultural, ecological, and environmental contexts. Phosphorus uptake. AMF benefits low-input systems by enhancing phosphorus uptake. High soil phosphorus levels can hinder AMF colonization, leading to copper deficiency in plants, as mycorrhizae facilitate copper uptake. Perennial cropping systems, cover crops in barren seasons promote AMF growth and hyphal networks, improving early season phosphorus nutrition and crop yields. Soil quality. Native AMF introduction expedites ecological restoration and soil recovery, enhancing soil aggregate stability through extraradical hyphae and glomalin-related soil proteins. Fluoremediation. Inoculating soils with AMF before vegetation reintroduction rejuvenates degraded soils, improving soil quality and extracting heavy metals. Rhizosphere ecology. AMF symbiosis influences the diversity and structure of soil species in the rhizosphere, impacting soil communities directly through exudates and indirectly via altered plant exudates. Global climate change. AMF dynamics and their interactions with plants are influenced by climate change, with varied responses to factors like drought, nitrogen deposition, and elevated carbon dioxide levels. Overall, AMF have multifaceted implications in agriculture, ecosystem restoration, soil quality, fluoremediation, rhizosphere ecology, and climate change adaptation. Ericoid mycorrhizae. Ericoid mycorrhizae represent a unique form of symbiotic relationship predominantly observed in members of the Ericaceae plant family. This association is established with a select group of mycorrhizal fungi, tailored to thrive in specific environmental conditions. Habitat and adaptation. Plants of the Ericaceae family, which form Ericoid mycorrhizae, are typically native to acidic and nutrient-deprived soils. These environments include boggy terrains, heathlands, and boreal forests. The formation of ericoid mycorrhizae is a vital adaptation that enables these plants to survive and flourish under such challenging conditions. Historical molecular clock analysis suggests that this symbiotic relationship originated approximately 140 million years ago. Plant families involved. While the Ericaceae family is a primary participant in this symbiotic relationship, other plant families, including the Epacridaceae and Empetraceae, also engage in ericoid mycorrhizal associations. This indicates a specialized adaptation shared among these families to cope with similar environmental challenges. Fungal partners. The fungal species Hymenociphes, Pezizella, Ericae was initially identified as the primary endosymbiont forming ericoid mycorrhizae with plants of the Ericaceae family. However, subsequent research has unveiled the involvement of other fungal genera, such as Audiodendron, Mixotrichium, and Gymnacilla, in establishing mycorrhizal associations with ericoid plants. Morphological characteristics. In terms of structural formation, ericoid mycorrhizae exhibit similarities with arbuscular mycorrhizae. The fungal hyphae penetrate the cortical cells of the host plants, particularly those of fine roots. Within these cells, the hyphae form intricate coils, akin to the arbuscules observed in arbuscular mycorrhizae. Notably, the fine roots of ericoid plants are characterized by a central vascular bundle surrounded by a singular layer of cortical or epidermal cells. In conclusion, ericoid mycorrhizae stand out as a specialized form of plant-fungal symbiosis, tailored to the unique challenges posed by acidic and nutrient-poor soils. This association underscores the adaptive strategies employed by both plants and fungi to optimize nutrient acquisition and survival in specific ecological niches. Orchid mycorrhiza. Orchid mycorrhizae represent a pinnacle of symbiotic relationships between plants and fungi, showcasing an intricate interdependence unparalleled in the realm of mycorrhizal interactions. With approximately 17,000 orchid species in existence, these plants heavily rely on their basidiomycete fungal counterparts for essential nutrient acquisition. Orchid fungal interdependency. The degree of reliance between orchids and their fungal partners is profound. In certain orchid species, the symbiotic relationship is so deeply ingrained that the orchid seeds carry fungal propagules. This ensures that the fungus is immediately available during seed germination, facilitating nutrient uptake during the critical early stages of plant growth. Significance in seed germination. Given the diminutive size of orchid seeds, they possess limited nutrient reserves due to the absence of an endosperm. Consequently, the initial development of the protocorm post-germination is contingent upon the symbiotic fungus, not just for nutrient acquisition but also for carbohydrate provision. This symbiotic reliance persists until the orchid develops its photosynthetic capabilities, a process that can span up to a year in certain species. Remarkably, some achlorophyllous orchid species never achieve this photosynthetic capacity, rendering their dependence on the fungal partner perpetual. Fungal structures, arbuscules, peletons within the cortical cells of the host orchid. The fungi form intricate, coil structures known as arbuscules or peletons. These structures are transient in nature, and upon reaching the end of their lifespan, they leave behind deposits of cellulose and pectin within the host cell. Intriguingly, new fungal hyphae can subsequently invade these cells, tapping into the stored carbohydrates and nutrients. In summation, orchid mycorrhizae exemplify the intricate balance and mutualism in plant-fungal symbioses. The profound dependency of orchids on their fungal partners underscores the evolutionary adaptations that have shaped these relationships, ensuring survival and thriving in diverse ecological niches. Monotropoid mycorrhiza. Monotropoid mycorrhizae are specialized mycorrhizal associations predominantly found in the Monotropody subfamily within the Ericaceae family. These plants exhibit a unique adaptation, having largely lost their photosynthetic capabilities and existing as achlorophyllous plants on forest floors. Initial misconceptions. Initially, plants associated with monotropoid mycorrhizae were believed to be parasitic on forest tree species. However, contemporary understanding reveals a more intricate relationship. These plants, rather than being direct parasites, leverage shared mycorrhizal symbionts common to neighboring trees. Carbohydrate acquisition via mycorrhizal bridges. Monotropoid plants obtain essential carbohydrates from adjacent trees. This transfer is facilitated through mycorrhizal bridges, which connect the root systems of the monotropoid plants and the neighboring trees. This intricate network ensures a continuous supply of photosynthetes to the achlorophyllous plants. Fungal partnerships. Several fungal species, including Tricoloma, Russula, and Rhizopogon, have been identified as partners in these mycorrhizal associations. On their monotropoid hosts, these fungi exhibit characteristics similar to ectomyocorisal associations on trees. They form fungal sheaths and heartig nets, which are typical of ectomyocorisal relationships. Additionally, these fungi produce unique structures called hyphal pegs, which originate from the inner sheath layer of hyphae and penetrate the tangential wall of the host's cortical cells. Each cortical cell typically houses a single peg. The formation of these pegs is closely tied to the host plant's growth and development, peaking during the flowering stage and subsequently diminishing. Nutrient transfer and acquisition. The pioneering work of B.J. Ockman in the 1960s utilizing radiotracers provided evidence for the transfer of photosynthetes from trees to monotropa plants. Further research corroborated that this transfer is mediated through mycorrhizal bridges formed by shared fungal hyphae. While the exact mechanisms of nutrient acquisition by monotropoid mycorrhizae remain to be fully elucidated, it is postulated that the process mirrors that of ectomyocorizal associations. In conclusion, monotropoid mycorrhizae exemplify the adaptability and complexity of symbiotic relationships in nature. Through shared fungal networks, these achlorophyllous plants have evolved a strategy to thrive in nutrient-poor forest floors, underscoring the intricate balance and mutualism inherent in ecological systems. Other types of mycorrhiza. There are other types of mycorrhiza such as arbutoid mycorrhizae and dark septate endophytes, DSE. Arbutoid mycorrhizae, also known as ectomycorism, are specialized symbiotic associations primarily found in specific Ericaceae plants like Arbutus and Arctostaphylos, and various Pyrolycae genera. These associations involve a variety of ectomycorrhizal fungi, forming delicate fungal sheaths around plant roots with a peripodermal heartig net and intracellular hyphal complexes. They facilitate nutrient and photosynthete exchange, potentially extending this interaction to neighboring tree species, emphasizing the interconnectedness of these relationships in ecosystems. Dark septate endophytes are root endophytes found in various plant species, particularly in cool and nutrient-poor environments. They exhibit taxonomic diversity, including genera like Chloridium, Leptodontidium, Phyalocephala, and Phyalophora. DSE fungi enter plant roots, forming runner hyphae and distinctive microsclerotia. Their functional role, still under investigation, likely involves nutrient acquisition, possibly through specialized enzymatic processes in nutrient-poor environments. DSEs contribute to plant health and ecosystem resilience, highlighting their significance in plant-fungal interactions. Function of mycorrhiza. Nutrient acquisition, central to the mycorrhizal function, is the enhancement of inorganic nutrient uptake from the soil. This is evident from numerous studies indicating superior growth in plants with mycorrhizal associations compared to those without. A landmark study by Ney and Tinker, employing radioactive phosphate, illuminated the superior phosphate uptake capability of mycorrhizal roots. The extensive hyphal network, with its vast exploratory capacity, facilitates efficient nutrient uptake, especially phosphorus. This nutrient acquisition mechanism is not only energy efficient but also genetically regulated, with specific proteins and transporters playing crucial roles. Carbon exchange. Mycorrhizae receive carbon in the form of photosynthetes from the host plant. This exchange is facilitated by the upregulation of hexose importer genes in both the plant and the fungus. This carbon exchange not only fuels fungal growth but also fortifies the mycorrhizal association against potential threats. Water acquisition. Mycorrhizal hyphae and rhizomorphs possess the ability to translocate water, a function particularly vital in arid environments. The efficiency of this process, however, is contingent on the plant species and environmental conditions. Plant defense mechanisms, mycorrhizae confer a dual defense to plants. Firstly, the physical barrier created by the ectomycorisal sheath offers protection against soil-borne pathogens and grazers. Secondly, the biochemical defense, mediated by fungal secondary metabolites, shields the plant against a spectrum of pathogens. This protective function is evident in agricultural settings where mycorrhizal associations mitigate the deleterious effects of pathogens, enhancing plant growth and yield. Disease prevention. Mycorrhizae play a pivotal role in disease prevention, especially in high-density agricultural settings conducive to pathogen proliferation. Their protective role extends to both fungal pathogens and nematodes. However, the extent of this defense in natural ecosystems remains an area of active research. Mycorrhizae and pollution. Pollutants, including acid rain, heavy metals, radionuclides, organic pollutants, and the effects of climate change, have significant impacts on mycorrhizal fungi. Acidifying pollutants, acid rain, containing sulfuric acid, affects mycorrhizae by reducing tree canopy photosynthesis, diminishing root energy supply, and increasing toxic metal availability in soil, disrupting mycorrhizal formation and nutrient uptake. Heavy metal pollutants, mycorrhizal fungi, especially ectomycorism, tolerate heavy metals, with certain species reducing metal uptake by host trees. Fungi adsorb metals onto hyphae, protecting trees from metal toxicity. Radionuclide pollutants, mycorrhizal fungi, notably ectomycorism, accumulate radionuclides and can enhance plant uptake while preventing translocation to shoots, acting as a protective mechanism. Organic pollutants. Some mycorrhizal species can decompose organic pollutants like PCBs, contributing to organic pollution mitigation. Climate change. Climate change factors, including elevated carbon dioxide, rising temperatures, and altered rainfall patterns, influence mycorrhizal communities, altering nutrient availability and community composition. Fungal conservation. Ectomycorrhizal species are declining due to pollutants like nitrogen deposition, raising concerns about fungal conservation. Efforts include habitat preservation and assessing the impact of mushroom harvesting on fungal decline. Overall, pollutants and climate change have complex and varied effects on mycorrhizal fungi, emphasizing the need for conservation efforts and further research to understand these interactions fully. Benefits of mycorrhiza. Mycorrhiza, the symbiotic association between fungi and plant roots, offers a wide range of benefits to both plants and ecosystems. These benefits are crucial for nutrient uptake, plant health, and overall ecosystem functioning. Here are some of the key advantages of mycorrhizal associations. Improved nutrient uptake. Mycorrhizal fungi extend their hyphae into the soil, increasing the root's capacity to absorb essential nutrients such as phosphorus, nitrogen, and micronutrients. They can access nutrient sources that are otherwise unavailable to plants, enhancing nutrient acquisition efficiency. Enhanced water uptake. Mycorrhizal networks can extend deep into the soil, accessing water in deeper layers during drought conditions, which helps plants maintain adequate hydration. Disease resistance. Mycorrhizal associations can enhance a plant's resistance to soil-borne pathogens by forming physical barriers between pathogens and roots. Some mycorrhizal fungi also produce antimicrobial compounds that inhibit the growth of harmful microorganisms. Improved plant growth. Mycorrhizal fungi stimulate plant growth by facilitating nutrient uptake and increasing the availability of water. Enhanced growth can lead to improved crop yields in agriculture and healthier, more robust plants in natural ecosystems. Stress tolerance. Mycorrhizal plants are often more resilient to environmental stressors such as drought, salinity, and heavy metal contamination, due to their improved nutrient and water uptake capabilities. Ecosystem restoration. Mycorrhizal fungi play a critical role in the restoration of degraded ecosystems. They help establish vegetation on poor soils, stabilize soil structure, and sequester carbon. Increased biodiversity. Mycorrhizal associations are associated with greater plant diversity, as they enable a wider range of plant species to thrive by accessing limited soil resources. Carbon sequestration. Mycorrhizal fungi store carbon in soil organic matter, contributing to carbon sequestration, which is essential for mitigating climate change. Support for non-photosynthetic plants. Mycorrhizal networks can connect different plant species, allowing non-photosynthetic plants, mycoheterotrophs, to obtain carbon and nutrients from nearby photosynthetic plants. Improved soil structure. Mycorrhizal fungi produce extracellular substances like glomalin, which help bind soil particles together, enhancing soil structure and preventing erosion. Increased ecosystem productivity. Mycorrhizal associations contribute to overall ecosystem productivity by promoting the growth of plants, which, in turn, support higher trophic levels. Reduced fertilizer dependency. In agriculture, mycorrhizal associations can reduce the need for synthetic fertilizers by enhancing nutrient uptake efficiency, potentially reducing production costs and environmental impacts. In summary, mycorrhizal associations are essential components of terrestrial ecosystems. Thank you for joining us in this insightful exploration of mycorrhizal fungi. If you're looking for comprehensive study notes related to this lecture video, simply head over to Google and search for microbiologynote.com. There, you'll find valuable resources to deepen your understanding of this fascinating topic. Remember, knowledge is a journey, and continuous learning is key. Stay curious and keep exploring the incredible world of microbiology.